Welcome to another AP Bio live review. I am really happy to be here, and I'm really happy to be doing my favorite thing, which is talking about AP Biology with motivated students who want to learn biology. I know that it's starting to feel kind of intense because the AP Bio exam is 2 weeks from tomorrow. But what I'm hoping is that through today's live review and through all the other things that I'm doing, we'll be able to relieve some of that anxiety and help put you on the pathway towards walking into the exam with confidence and walking out feeling that you crushed it, because that is the goal of what we're doing here. So I'm really happy to hear that people are here in the audience, and I'm really happy to see this comment by Muhammad, who is telling me that it's 2:48 a.m. You are a superhero, Muhammad. Way to go. And I am so happy to be here and so proud of you for showing the kind of commitment that's needed to be shown to do what needs to be done to succeed on the AP Bio exam. So Muhammad, good for you. I'm going to change my view a little bit so that I look a little bit more like, no, that's not what I want. Yeah, that's good. Maybe even a little bit more like, no, that was better before. Let's go back to this. Okay, so holy cow, we've got so many folks here who are chatting in. And in fact, I really want to encourage you to do that, because knowing that there are real people on the other end means so much to me. So I've got people who are telling me that it's 3 a.m. where they are. Oh my goodness. And Unaya is saying that your first final for AP Bio, the short FRQ, is here. And well, there we are. Anyhow, I am so honored that you've chosen to spend your Sunday evening, or maybe it's Monday morning for some of you, like Abdallah and Muhammad and stuff like that. So anyhow, here you are, and let's get right to it. Let me tell you what the plan is for today. And I'm just scanning the comments, and I really want to encourage you to do these comments, because first of all, what's cool about YouTube Live? The fact that you can ask questions and I can respond. So let's go. I'm also assuming that my audio is fine. I kind of have a good confirmation myself that my video is fine, but why doesn't somebody just chat in and say, we can hear you, Mr. W, just fine. That is always a big confidence builder for me, because there we go. Fantastic. And wow, that's fantastic. And I'm seeing a comment from Asba, who's saying that they have been studying for all of spring break, and that's really what needs to be done. So guys, let's go. Let's talk about what the plan is for tonight. Here's our agenda. I want to talk a little bit about how to review for the AP Bio exam. I've already done a presentation about that, and if you go to the Science Music Videos channel, you'll see I have 2 videos. One's called like Your Plan for a 5, and the other was like a day-by-day plan, and that tells you everything. And so you can spend 20 minutes with those videos, and I don't want to redo that right now. So I just want to let you know that how to review is something I've covered. It's on the Science Music Videos channel. It's waiting for you. And I also have a downloadable plan. Let's see if I can find a banner for that. Yeah, there we go. So if you go to apbiosuccess.com/plan, you'll get a day-by-day plan for 2 weeks. And it actually kind of starts today. So go ahead and do that. I'm going to take that down right now, give myself the whole screen, and let's go on. So we're going to talk a little bit about that. I thought that, we got to pick and choose about what we're going to talk about tonight. I could talk about everything, but we can't do that. So we got to make some choices. So what I thought I would talk about is mostly stuff related to topics 1 and 2. And if people can let me know, if you could make a webinar at about this time tomorrow night, like at about 5 p.m., Pacific time, same time as I started. Can you just in the chat let me know? Yeah, that would work for me. Okay. And so let me see. Anybody, can you make it tomorrow at 5 p.m. Pacific time? Because if I get like even 10 people to say yes, then I'll just set up another show. And Muhammad can, that's cool. That means you're going to be staying up really late. And Hassani can, and Chloe can. Let's just get a couple. Okay, so fantastic. So I'm seeing a bunch of people who are saying that works, and that's really good. Alnia, I hope I'm saying your name correctly. Yeah, this goes onto the live tab on the Science Music Videos channel, so you can go and you can watch these. They live up there forever. Sometimes I even edit them and re-release them, but I don't think I'm going to be able to do that. So yeah, you can see this. So anyhow, what I thought I would talk about tonight were proteins, membrane transport, and the diffusion osmosis labs. People are constantly asking me, like, what do I need to know about the labs? Some of the labs you don't need to know that much about because they're difficult to replicate. But these are labs that actually you do in AP Bio classes, and they actually work. And so I thought it would be useful to go over them because they also do a good job in helping you understand about diffusion and osmosis. Now, I will change that plan if people have specific questions, but I want to keep it to 1 and 2 tonight. Oh, and then I was just going to go on. And then Unit 3 we'll do, we could do tomorrow night. All right. And then I'll post a schedule for doing the rest of these units. And I also want to let people know about something. I know that not everyone is available and can do it, but I want to just let everybody know about it, that I am doing also reviews on Zoom, and they're much more intimate and much more interactive than this one is because, you know, Zoom, you get to really talk back and forth and you can ask questions. Those are paid events, but you're welcome to join us. And I'm going to be doing one on Tuesday at 5 p.m. And then I'm doing one the following Sunday. If you go to this URL, apbiosuccess.com/exam review, you can see the schedule and you can see a sign-up sheet. And I want to invite you to come, but I want to emphasize, unlike this event, which is of course free, here you are, and I'm very happy about that. These are paid events, but I'm hoping you're going to be there. So apbiosuccess.com/exam review. Now, I'm also hoping that if we have time, we can do a couple of practice FRQs. So we're going to start with how to review for the AP Bio exam. After that, I'll open it up for questions. You can tell me if there are any specific topics you want to review. And if there aren't specific topics, we'll start by talking about proteins, membrane transport, and diffusion osmosis labs. So you got 2 weeks from really, I mean, you know, this time 2 weeks from now, your studying is going to be pretty much coming to an end. And that's actually an important point that I want to say a little bit. Do this right. Don't leave your studying for the weekend before the exam. You know, if you were a gardener, you plant your seeds way ahead of harvest time. Studying is like that. You've got enough time. If you start now and you follow the plan that I had the banner up for before, it's this plan over here. You follow this plan, you're going to be okay. You're going to be able to do a nice, slow, thoughtful, careful review, and you won't have this terrible experience the weekend before the exam. So do the work, all right, and start it now, and, you know, be mature about this. And I'm seeing Charlotte saying that she's been studying for at least an hour every day, and that's exactly what you got to do. And I'm just going to respond to a question right now from Asba. Functional groups, you don't need to know them. Like, you don't need to say, oh, that's methyl, that's hydroxyl, stuff like that. But what you need to be able to do is know that, like, a hydroxyl group, an OH attached to a molecule, makes a molecule polar. So, like, therefore, glucose, which has a lot of hydroxyl groups, is polar and it dissolves in water. So that's the kind of thing. And Aisha is saying, if I haven't learned anything throughout the year, is a way I can get a 4 or 5 by studying the last 2 weeks. I think that in 2 weeks of studying, but like Aisha, you got to really show up. You got to go like 3 hours a day, every single day. You've got to use interactive resources. I offer many of these on my website. So I'm going to encourage you to subscribe to my website because you won't have to do all the preparatory work. And I'd also suggest that you come to my review webinars if you can. Because if you haven't learned anything throughout the year, and I hope that's not on account of, I mean, I'm sorry for that. I don't want to throw your teacher under the bus, but who knows what happened to cause that, but that's not really what we're going to talk about now. Let's talk about the goals of AP Bio Review. All right, goals of AP Bio Review are as follows. You don't have to like learn the whole course unless you're in a position like Aisha just mentioned where you haven't learned anything. What you want to do is you've already learned a tremendous amount. You've been taking tests, you've been taking quizzes, stuff like that. So what you have to do is you have to find the gaps in your knowledge and fill them. So that should be the guiding question that you are reviewing. What are the gaps in my knowledge? How do I fill them? And then the way that you fill them is through authentic practice. And that means you're going to be doing a lot of MCQs and FRQs. And I would add to that, even though it's not on the slide, flashcards. And the goal is that you become confident, skilled, and fluent. In other words, you walk into the test feeling like I have studied really well and I understand the basic principles that needed to be learned in AP biology. And then you can write about that with fluency when you're doing FRQs on Monday the 5th. And when you're looking at the multiple choice questions, you're like, oh, I'm familiar with this material. That's how you want to set yourself up. Is that an easy road to get there? No. But the fact that all of you are here and you're here 2 weeks ahead of time, which means that you're thinking about this in a really smart way, that means that you're positioned to be set up for success. So what's the best way to review? Let me emphasize, first of all, what's not a good way to review. And this is going to sound kind of weird because I'm a big video maker on YouTube. But like just watching videos in and of itself is not such a good way to review because it's passive. You like feel smart when you watch a video, like you understand what I'm saying and you feel like, oh, I get it. That's not really the right way to review because you need to have your knowledge at a much, much higher level. So the best way to review does not involve simply watching videos. That can be a start. It doesn't involve rereading your textbook. It doesn't involve highlighting stuff. It doesn't involve rereading outlines and things like that. That is not the best way to review. The best way to review is a method that's called retrieval practice, accompanied by spacing and interleaving content. I'm going to quickly go over what those three things are. Retrieval practice is also known as the testing effect. And what it is it's actively recalling information from memory. In other words, it's not reading what a phosphorylation cascade is about. It's about asking yourself the question, how does a phosphorylation cascade work? And if you can't talk about that with some fluency, then you make a note to yourself and you say, I need to learn about phosphorylation cascades. I need to learn about cell communication. And you want to do that with every topic in the curriculum. So how do you do retrieval practice? I'll show you in a minute, but why? It strengthens memory and recall. You're going to walk into that test, you've got to recreate the knowledge. You're going to have to write FRQs. So how do you do that? Flashcards, interactive outlines, where you ask yourself questions from an outline and then generate the answer. Interactive FRQs, which we have on my website, learn-biology.com, and then practice tests or real tests. And we also have many, many hundreds of practice multiple choice questions on learn-biology.com. And again, I'm going to emphasize the importance of using an interactive website like mine because we've done the work of preparing for you. And all you have to do is put the time in and learn. Spacing effect. This is spreading study sessions out over time. Don't cram. Start now. Learn stuff. Allow yourself a little bit of time to forget it and then learn it again. So spread out study sessions over time. It improves long-term retention because your brain has to work a little bit harder and you got to schedule it. How are you going to schedule it? You're going to schedule it with this. I've scheduled it for you with my apbiosuccess.com/plan. Download that plan if you haven't done it already. Let's go on. Interleaving content is something that's very counterintuitive for students. What you want to do when you're studying for a big test like the AP Bio exam is mix different topics and types of problems into a study session as opposed to topic by topic studying. You're motivated. That's why you're here. And you're probably thinking like, okay, I'm going to be organized. I'm going to do AP Unit 1, then I'm going to do AP Unit 2, then I'm going to do AP Unit 3. That's not the way to do it. In your study sessions, you want to mix up different topics. And doing that, asking yourself a question from Unit 1, followed by one from Unit 5, followed by Unit 3, followed by Unit 8, that builds connections between topics. It leads to more flexible thinking and better problem solving. And how do you do that? You do that with my website, learn-biology.com. And learn-biology.com is College Board approved. It's got built-in retrieval practice, spaced repetition, interleaving. It's got flashcards, multiple choice questions, and enhanced free response questions with personalized AI feedback. And it includes access to the Biomania app. So how do you get learn-biology.com? You're going to download it by going to where? Here. Just go to learn-biology.com/apbiology. There's a sign-up sheet and you can sign up. Okay, so let's go further. I'm going to skip that. Here's a 3-step plan. You got to self-assess to find your gaps. How are you going to do that? You're going to find a checklist, like maybe your teacher has an outline. If your teacher didn't give you a good outline, I've got a great outline that you can use, and that is on apbiosuccess.com/checklist. Just go download that. You can take tests on learn-biology.com. That'll give you further information. Then you're going to use retrieval practice to fill in the gaps. You're going to use flashcards. You're going to use our video tutorials, which have videos followed by summaries, followed by flashcards, followed by quizzes. And you can also consult your textbook, but you've got to be interactive about it. You've got to make flashcards. You've got to do something that makes you retrieve the information. And then here's my number one tip. Spend a lot of time over the next 2 weeks writing FRQs. The more you do that, the better you're going to do on the test. You can write them from the College Board. The College Board gives them all away. You've got 2024, 2023, 2022, going all the way back to like 2008. So just search for past AP Bio FRQs in Google. You'll go right to the page. And what we offer on learn-biology.com are enhanced FRQs where you actually type in a response and you'll get feedback. And I'll show you some of those later. So again, I got this all in my 2-week plan, and that's it. So I haven't looked at the comments for a little while, and they've been accumulating. So, okay. So I would just say, like, responding to Asba, if you take notes from a video online, then use a Cornell Notes format where you take your notes and then you put sort of headings on the left column and then you study from that. And then the way that you study is you force yourself to recall the information. So that would work. I can save you that effort through the flashcards. And I'm seeing some other techniques. And Audrina, Calvin cycle, we're going to talk about tomorrow night. And you don't need to know, I can tell you tomorrow night exactly what you need to know. And Lolipo is saying, what's the best way to study? I'm just going to break away from this and I'm going to go here. So what I would do if I had a test is I would go to learn-biology.com. All right, this is my website. And I would go to AP Bio Review. And I would, am I not signed in? Okay, I'm signed in, so great. And I would get started. So I would like go to the multiple choice review. And, you know, I talked about interleaving before. So I'm going to make this a little bit bigger so you can see. And I would start asking myself questions. Okay. And here's a question: in a marine food chain that ends with a humpback whale. And anyhow, I'm not going to, let me just show you this. I'm just going to do a little trick here. Page. You can't do what I'm doing because you're not the programmer of this thing, but like the next question is about cell communication. The next question is about population growth. The next question after that is about enzymes and energy. So this is what I would do, Lalipo, right now. I would go to learn-biology.com, I'd sign up, I'd start doing this. Then after I did multiple choice questions, I would go here and I would go to our free response questions. And I would do the same thing. I do my settings so that I get questions from everywhere. I'm going to begin practice. Okay. Figures A, B, and C below represent stages of cell division from the same organism. One shows the first division of meiosis, one shows the second division of meiosis, and one shows division by mitosis. Identify which figure shows an event for mitosis and which figure shows an event for meiosis 1, which figure shows an event for meiosis 2. Justify your choices. So I am looking at figure C, and I'm seeing that homologous pairs are paired up. So I'm going to say, so part 1, figure C shows meiosis 1 because you don't really have to worry about the spelling. Homologous pairs are paired up. Now I'm going to make a deliberate error. I'm going to say figure A shows meiosis 1 and figure B shows mitosis. All right. So now I'm just going to hit check my answer. Okay. And here's what I want you to know. I hope you can see this. I hope it's big enough and make it a little bigger. Your identification of figures C, A, and B is incorrect. According to the answer guide, figure A is mitosis. Figure C is meiosis 1. Ah, right. And figure B is meiosis 2. Your justification for figure C, meiosis 1, is correct. Homologous pairs are paired up. I'm not going to go any further, but I'm going to show you, I mean, basically what I hope I've shown you, and this is for everybody, is that you're going to get feedback as if you had me tutoring you. You know, I actually wrote these answers and I helped program the AI. So if you do these enhanced free response questions, you're going to get marvelous feedback. So Lalipo, that's what I would do. I'd sign up for learn-biology.com tonight, and I would just test myself, because that's how you're going to learn. All right, let's go back to our presentation. Almost there. Yeah, okay. So now that we are there, so I had some topics that I was just going to like start teaching, but I am wondering about this. I'm going to do Unit 3 tomorrow. So tonight, you've got this list of topics for AP Bio Units 1 and 2. If you've got a question, if you want me to go over any of these, put it in the chat. And I'm seeing Hassini is actually saying osmosis is confusing. And any other topics that people want me to do? Just I can start with osmosis, or my plan was to do proteins and then to do membrane transport and then to do labs. Anybody have any preference? Number 9, cell size. Okay, I could definitely do that. So I'm just going to make a little note. We got osmosis. And we've got, number 9 is cell compartmentalization. Okay, I can do that. Okay, let's do it. Osmosis first, then cellular compartmentalization. Let's go. All right, so let me find the slide. here. Okay, beautiful. Osmosis and its consequences. And I'm seeing some more responses. And jellyfish. I talked about this tomorrow. I'm going to do unit 3 tomorrow. All right. So osmosis. What is it? You need a little sip of tea. Osmosis is the diffusion of water. So everything on a molecular level diffuses from high concentration to low concentration. That's just based on the random movement of molecules. And water does the same thing. But because water is the solvent of life, there's some connected terminology that's a little confusing that we have to talk about. So here we have a situation where we have a body of water that's been subdivided into two by a selectively permeable membrane. And the membrane lets water diffuse through, but not the solute. And the solute's represented by these little red spheres. The water concentration is higher over here than it is over here. Why? Because this might be 99% water. It's just got these two molecules of solute floating around. Whereas this might be 90% water. And I said two molecules, but that was a ridiculous thing for me to say. You know, 2 parts versus 1, 2, 3, 4, 5, 6, 7, 8, 9 parts, because that's what these red spheres actually represent. So there's relatively more water here and relatively less water here. So water is going to diffuse through this selectively permeable membrane from where it was more concentrated to where it's less concentrated. That's the basic idea. Now let's start throwing in all of the difficult terminology. The terms hypotonic and hypertonic are relative terms. Hypotonic means that there's relatively more water. So over here we have a lot of solute. Over here we have very little solute. So this is the hypotonic side. This is the hypertonic side. Hypertonic means relatively less water, relatively more solvent. And what you have to remember, what you have to memorize, is that water diffuses from hypotonic to hypertonic. Say that over and over again. Say it in your sleep, because that's how it works. Hypotonic to hypertonic. Water is going to diffuse from where it's more concentrated to where it's less concentrated, from where it's more concentrated to where it's less concentrated. This is a device that's called a U-tube, with a U, not a Y-O-U, like the platform that we're on right now. And what we're doing is we're adding some solute to water over here. This is a selectively permeable membrane. Now, this side is hypotonic. This side is hypertonic. And the water level on the left side, where we added the solute, the sugar, is going to rise. Why? Because water always flows from hypotonic to hypertonic. Let's skip the slide. Here's a cute little lab that I actually have a song about. I have a song that's called Osmosis, and it's about gummy bears, or at least it starts talking about gummy bears. If you put a gummy bear in water, it will expand. Why? The gummy bear is mostly sugar. Water in the bowl is 100% water. Water will flow from the hypotonic solution in the bowl to the hypertonic gummy bear. That'll cause the gummy bear to expand. Why? Because water flowed from hypotonic to hypertonic. I'm not going to do that because there's some problem with the file. Sorry. Next slide. Okay. Where it gets a little confusing is when we start looking at cells and how cells respond to hypotonic and hypertonic environments. In this slide over here, the cell had to have been hypotonic to its hypertonic environment. Therefore, water flowed out of the cell into the hypertonic environment because water always flows from hypotonic to hypertonic. In this situation, the cell is isotonic to its environment. Water is entering and leaving at the same rate. In this condition, the cell is hypertonic to its hypotonic environment, so water is flowing into the cell. And I'll just show you those 3 slides, and you can read them while I take another sip of throat coat tea. In my Science Music Videos cup, which you can buy on the Science Music Videos channel if you want to, but really that's not the important thing. What I do want you to do is I really want you to subscribe to learn-biology.com because it's going to completely transform your experience studying for the AP Bio exam. Okay, how are we doing over here? Looking, just scanning some of the questions. All right, let's go on with osmosis. This is a very commonly observed organism in AP biology classes. It's a plant. It's called Elodea, and it's kind of algae, and you can just pull the leaves off and lay them down on a microscope slide. In the left, Elodea is in its normal freshwater hypotonic environment. But if you put it in a hypertonic environment, like a very salty environment, water will leave the cells and it'll flow into the environment. That will cause the membrane to peel away from the wall. This process is called plasmolysis, but that's not very important. But the main thing is that if a cell looks like this, a plant cell looks like this, that's because it was placed in a hypertonic environment. And that's what a salty environment can be like. The salt makes the membranes, the salt makes the cells hypotonic to their environment. Water leaves the cell, the membrane peels away from the cell. This is also known as plasmolysis. Animal cells, a little bit different because they don't have a wall, but the basic principles are the same. Here, the cell was in a the cell was hypotonic to its hypertonic environment. Water leaves the cell and the cell shrivels up. In the middle, the cell's isotonic to its environment. Water enters and leaves the cells at the same rate. This is what you have to do if you want to keep cells alive in culture. They need to be in an isotonic environment. And on the right side, the cell is hypertonic to its watery environment. Water enters the cells and the cells actually burst. Again, I explain all this in my osmosis song. And I also have a video that you can watch on learn-biology.com. This is a cool thing. This is a freshwater protists. Protists are eukaryotes that are single-celled. It's not a good evolutionary category, but it's very useful. And basically, protists in freshwater are hypertonic to their freshwater environment. So imagine this living in a pond or a lake. Water would flow from the pond or the lake into the paramecium because the paramecium has all of this stuff dissolved in its cytoplasm. In order to stay alive, the paramecium has this special organelle that you don't have. It's called a contractile vacuole. And what it does is it pumps, as you can see in this video. And as it pumps, you can't see the water being expelled, but what it's doing is it's expelling water. And this is called osmoregulation. This is how the cell is maintaining its osmotic environment. Here's a cool graph. This has actually shown up on FRQs, and here's how you'd have to understand it. As you increase the molarity of the solution, the rate of contractile vacuole contraction drops. Why is that? Because as the environment becomes more hypertonic, there's less of a concentration gradient causing water to flow in to the paramecium's cell. So therefore, it doesn't have to contract as frequently. In pure water, that contractile vacuole has to work hard to pump water out. But as the solution that the paramecium is approaches the osmolarity of the cytoplasm, the contractile vacuole would have to contract less. All right, so that's a cool application. Let's see if this video is working. Yeah, blood cells placed in a hypotonic solution, what's going to happen to them? The cells are hypertonic to the watery environment. So you can predict that they will, here it goes, they're going to burst. The cells explode. Why? Because water is flowing into the hypertonic cells. All right. So anyhow, that was a review of osmosis. There were other things that people wanted to review. Let me look. So that was osmosis. And then the next topic that people wanted to review was about cell compartmentalization. Just want to double check. That's what I wrote down. Okay. Wait, do I have a slide about compartmentalization? Yeah, well, this is a pretty good slide about cellular compartmentalization. So there's a system within eukaryotic cells. It's called the endomembrane system. And it's a system of internal membranes that divides the cell up into different compartments. Why would a cell want to do this? For a couple of reasons. First of all, if you think about a structure like a lysosome down over here, the lysosome is used to digest things. It often is highly acidic and it's full of hydrolytic enzymes. Hydrolytic enzymes do hydrolysis, and that's breaking polymers into monomers. These enzymes are, in a way, quite dangerous. They can't be in the cytoplasm, or else they would wind up digesting the cytoplasm. So by having a compartment, the cell can put these dangerous enzymes into this compartment, segregating them from the rest of the cell. So one thing that cellular compartmentalization does is it allows for separation of functions. The second thing is that if you see over here, the rough ER, the rough ER is rough because it has ribosomes attached to it. And the fact that there's a rough ER provides a huge amount of surface area that a cell can use when it has to construct things that are going to be embedded in a vesicle, exported from the cell, or head to a lysosome. So increase in surface area through compartmentalization, and also like working space for membrane embedded enzymes. So that's the why of compartmentalization. The thing to know is that compartmentalization is pretty much a eukaryotic feature. So the three eukaryotic kingdoms, fungi, plants, animals, they're all highly compartmentalized. And then I was talking about a paramecium before. That's a eukaryote too, and it also has many cellular compartments. And again, think about like a luggage compartment on a plane. It's a place to put things. So that's what compartmentalization does. Prokaryotic cells are small, single-celled cousins. They are not compartmentalized. There might be a little bit of compartmentalization, but you want to connect that in your mind with eukaryotes. Now, I have a slide coming up about, where is it? Yes. Okay, so compartmentalization is really connected with, I think I have a slide about this. Sorry, I'm going to go like this and find it. Oh, here it is. Okay. Oh, sorry. Okay, so this was the slide I wanted to find before. This is about the endomembrane system, and this shows all the different cellular compartments that you should know. And how did these compartments arise? At one point, about 1.8 billion years ago, eukaryotic cells arose through a fusion of two prokaryotic cells. There are three domains: archaea, bacteria, and eukarya. And the eukarya were formed by a fusion of an archaeal cell and a bacterial cell. How that fusion happened, it's not so important for you to know. The diagram that you've learned might look different from this, but the whole point is that a bacterial cell found its way inside of an archaeal cell, and that bacterial cell evolved into a mitochondrion. This is the endosymbiotic theory. And it's important to know because you might be asked a question on an FRQ that would be something like, what's the evidence for the idea that mitochondria were once free-living cells? And you'd have to say three things. Chloroplasts arose in a similar way. They were engulfed, but by an already eukaryotic cell containing mitochondria. And here's the evidence. The evidence is several-fold. And this is something you should know because you could get this on an FRQ. So like know this from memory. Both chloroplasts and bacteria and mitochondria, have their own circular DNA. That's the kind of DNA that we find in bacteria and archaea. Our DNA, our eukaryotic DNA, is linear. They replicate themselves through binary fission. In other words, the mitochondria grows and then it splits itself in half as a bacteria does. It doesn't do mitosis the way eukaryotic cells do. These organelles have their own ribosomes and they produce some of their own proteins. Many of their proteins are produced by our cells, which shuttle those proteins over to the mitochondria through the endomembrane system. But they also make some of their own proteins, and their ribosomes are like bacterial ribosomes. And they also always have a double membrane. Sometimes they have more, and that's a sign of the fact that they were engulfed or taken up by a bacterial cell. So I am looking at the chat, and I'm seeing so many wonderful comments. And there's a request for water potential. I can do that. And I just want to clarify something. I'm seeing a question from Hallow. Nothing is hypotonic or hypertonic without reference to something that it's adjacent to. So you can have a hypotonic solution that would be adjacent to a hypertonic solution. But nothing can be hypotonic or hypertonic in and of itself. And you have to read the question carefully because they're going to specify whether the thing that's hypotonic is the environment or the cell. There's not a rule for that. And Legend Fire asks what unit we're talking about. This is all stuff in Unit 2. And Babbitt is just saying something very nice, which is, I don't know what I would do without your unit review videos. And thanks. I'm so glad that those are useful. And Audrina, I haven't really posted a schedule for the live stream. I'm going to try and do it, and then I'll put it on the community tab in YouTube, and I'll email it to folks who are on my mailing list because they signed up for something related to learn-biology.com. Okay. And jellyfish tomorrow for photosynthesis. Okay. You know what? I'm going to sort of rerun the clock a little bit, and I'm going to go like this. Okay. Can people do this now? Like, I'm sort of losing track of what people want. I think there was a request for Cupid is asking me to do another thing for hypotonic for describing a solution. And somebody asked about water potential. So why don't I do water potential? I'll do that. I'll jump back to osmosis and we'll do water potential. Okay, finding osmosis again. Okay. Oh, water potential, fantastic. Okay. All right, beautiful. So the question was water potential and another request for water potential. And Cupid, could you do an example of using hypotonic and hypertonic for describing a solution? Sure. We're talking about water potential now, but I'll start by referencing water potential after I take another sip of tea. All right. You have this device called a U-tube membrane that can let water pass through, but not the solute. What's the solute? Sugar. We're pouring in sugar to the left side of this U-tube. All the sugar is going to stay on the left side because it can't diffuse through this. By adding this solute to the left side, we've created a situation where the left side is hypertonic. It's got more stuff dissolved. That's what hypertonic means. And the right side is hypotonic. It's got less, I hope I said that the right way. This is hypertonic. It's got more stuff dissolved. And this is hypotonic. It's got less stuff dissolved. So that's what I meant to say. If I said it wrong the first time, I'm sorry. But hypertonic, hypotonic, more dissolved solute, less dissolved solute. And remember, say this in your sleep, water always moves from hypotonic to hypertonic. Now, how does that relate to water potential? Water potential takes this kind of intuitive thing related to osmosis, hypotonic, hypertonic, and it makes it much more mathematical. Water potential, which is represented by this Greek letter psi. And by the way, like there are equations connected with water potential. They're on your formula sheet. There's one connected to calculating solute potential. And I'm just going to bet that, I mean, this might be a bad bet. It's not worth me teaching it. I do have a tutorial about it. You can check it out. Do I teach that? I don't even know if I teach it. I think you need to know the basic water potential equation, and you need to know what I'm about to teach. I don't think you have to go deeper. So water potential, psi, it's a measurement of water's tendency to move from where it is to where it's not. Adding solute decreases water potential. Adding pressure increases water potential. So like, for example, if you press on a syringe, the water goes out. Why? Because the water potential is higher here than it is outside. If you add solute, like sugar, water moves from higher water potential to lower water potential. Why? Because adding solute lowers the water potential. What is the water potential formula? You got to remember this rule. Water always flows from areas of higher water potential to lower water potential. And here's the formula. Water potential equals solute potential plus pressure potential. Adding solute decreases water potential. Adding pressure increases water potential. And again, you don't have to memorize this. You can just find it on the formula sheet. And for those of you who are like worried and you're like, what? I just want to show you something for a sec. Let's go like this. And now let's go. So I'm going to my website, learn-biology.com. I'm going to courses and I'm going to the AP Bio video pathway. I'm going to make this a little bit smaller. And I'm just going to scroll down. And I'm in Unit 2, and there's a tutorial about osmosis and water potential. And what do you have there? You have my video about osmosis. You have my video about water potential. You've got my song about osmosis. You've got a summary of osmosis and water potential. You've got flashcards. You've got quizzes. So again, like if you're feeling like some of you are like, I don't understand water potential, I really need a review, sign up for learn-biology.com. And you do that through this. Just go to learn-biology.com/apbiology, just sign up. And then you'll have access to, I mean, the videos are on YouTube, but you'll have access to the flashcards. You'll have interaction. You'll have access to the interactive piece, and it's the interactive piece that makes you learn. Back to my explanation of water potential. And let's go here. And let's go. No, that's not what I wanted. I want this. I want this. Whew, good. Okay. Back where I want it to be. Let's go to this view. Where are my slides? No, that's not right. Sorry. Sorry. Brought up the wrong thing. What I wanted was this. Sorry about that. Okay. Here we are. Let's go right back to water potential. Okay. Now let's get into the numbers. Okay, so we talked about the formula. So now let's explain this using water potential. When we added the solute, what we did was we lowered the pressure potential. Water potential is measured in units called megapascals. You don't really need to know that. You just need to know that there's a unit for measuring water potential, MPa, and that if by adding solute, we lowered the water potential because there's no pressure. It's an open U-tube. We've lowered the solute potential. So we're going to take our pressure potential, and here's our solute potential. Now our total water potential is minus 2,3. The water potential on this side is 0. It's just an open container. There's no pressure. There's no solute. The water potential here is higher than it is here. Water moves from higher water potential to lower water potential. That's it. It's not more complicated than that. And let's look at another more complicated example. I hope that your teacher had you do the diffusion and osmosis labs. Those diffusion and osmosis labs are some of the best labs in the curriculum, and they actually work. If you put potato tissue into a beaker and you leave it overnight, the cells will expand, and you can actually mass them, and you can see how much they gain mass. And you can do a lot of cool things. You can just put them in pure water, and you can also put them in solutions of various molarity. Sugar solutions is what people use because sugar is cheap and easy to get. So if you put the cell in water, there's no pressure potential, right? But there's solute potential in the cell. It's -1. The water is going to flow into the cell because the solute potential is 0 here. The pressure potential is 0. The overall water potential is 0. Water is going to flow from the beaker into the cell. The cell is going to expand and gain mass, but not forever. Why? At a certain point, the wall, plant cells have walls, potato is a plant, is going to resist the pressure. So at a certain point, the pressure potential will rise to 1. The solute potential is already -1. That'll zero out, so the cell will be at equilibrium. Again, there's a lot here. If this is complex, please go to learn-biology.com, do the tutorial about water potential, watch the video, and you'll be fine. And let me see if I can answer these questions. Is water potential always negative or 0? No. I'm looking at Audrina's question. If you had a syringe and you pressed on the syringe, if you had water in a straw and you closed the end of the straw and you blew into the straw, you'd be adding pressure. Now, if you suddenly release the end, the water would squirt out. Why? Because water would go from higher water potential to lower water potential. So it's not always 0. And Priyanthan, I don't have too much more to say about pressure. If you squeeze on something, you create pressure, and that's it. And Ronito is answering questions about the electron transport chain. Good for you. I'm going to talk about that tomorrow, okay? So I'll just announce this right now. Tomorrow, either 5 or 6 p.m., I'll put a notice. in the community tab on YouTube, and I'll let you all know. All right, so that's the deal. All right, so we did water potential. Milo, diversity index we're going to do later. Small bites, you're a big fan, and I love that. That's so great. Okay, so we did water potential. We were going to do something else. So here's what I'm going to do again. What time is it? Jesus. Excuse me. Easter Sunday, I swore. All right. We've got time for like one or two more topics. All right, tropical vibes, not tonight. I'm going to do that later on. And I also want everybody to know if you check out this schedule, these are not free lives like this is. These are paid events, but I've got that whole thing organized. I haven't organized the YouTube lives quite as much, but I will. It's time. It's time for me to do that. So, number 7, sickle cell anemia. Yeah, perfect. Okay, so let's do that. And actually, before I do sickle cell anemia, I'm going to do one of the things that I really wanted to do tonight, which was proteins. So let's jump to proteins. Let's go like this. Sorry. Let's go like this. Okay. So, proteins. Super important to know about proteins because they're the action molecules of life. They're the molecules that get things done. How do you move? How do you move? How do you contract a muscle? It's protein. How do you do chemical reactions? It's protein. Our skin, protein. Our hair, your hair, protein. How do we transport things? Proteins. Sometimes we do energy storage with proteins, like the white of an egg. It's protein. And transmitting signals. Many hormones, like insulin, are proteins. Monomers are amino acids, and you should know amino acid structure. Central carbon at 1, there's an amine group, that's this over here. And if you have trouble remembering that, I don't know if this will be useful unless you have chemistry, but like ammonia is NH3, this is an amine group. There's a hydrogen atom, there's a variable group over here, and that variable group is also called a side chain, and it can be polar, nonpolar, acidic, or basic. There are 20 amino acids. Every living thing is made from the same 20 amino acids. And somebody asked me before, like, do you need to know functional groups? No, not really, but it's certainly useful to know that, like, if you stick this CH3, that's nonpolar. So this would make this amino acid relatively nonpolar. Whereas this functional group, a hydroxyl group, that's polar. And how do you tell if a side chain is polar, nonpolar? So tropical vibes, I hope I'm telling you this right now. If you have a carboxyl group over here, so you see that negative charge over there, that makes this acidic. And if you have the NH3 over here, that makes it basic. So again, this is sort of a mixed message on the functional groups. I have a video about functional groups, a tutorial about that too. I don't know if now is the time to know that in depth, but it's really useful throughout the course to know those. There are 4 levels of protein structure. There's the primary structure. That's what you inherit. It's spelled out in your DNA. There are interactions involving the polypeptide backbone. And that is the one amino acid connected to the next, that makes up a polypeptide backbone, and interactions that happen within the polypeptide backbone are secondary structure. Tertiary structure is this over here. It's interactions between R groups, And finally, quaternary structure is multiple polypeptide chains. Primary structure, like I said, it's genetically determined. It's a sequence of amino acids. The bond between amino acids is called a peptide bond, and that's one of the few bonds that you kind of need to know by name. Like you need to know hydrogen bonds when you see them. You need to know covalent bonds in general, and this is a covalent bond, but you should also know that the bond that connects amino acids is a peptide bond. And peptide bonds are catalyzed by ribosomes during protein synthesis. That's a topic in Unit 6. I'll put the schedule up. I know I haven't done that. Sorry. All right. Secondary protein structure. Interactions between the polypeptide backbones. So like, for example, carbon, carbon, nitrogen, carbon, carbon, nitrogen. That's the backbone. And the interactions between these functional groups are what cause there to be two forms. And you don't really need to be inside this. You just need to be able to recognize it. You need to be able to recognize that like this is called an alpha helix. Oops. And this is called a beta pleated sheet. The alpha helix is sort of one polypeptide backbone that's forming a corkscrew. This happens when two polypeptide backbones become adjacent to one another, either like this or like this. Okay, so that's how you get these beta pleated sheets. Tertiary structure is interactions between those side chains, and those can be hydrogen bonds. That's what we got going on over here. You can have covalent bonds over here between this sulfur atom and this sulfur atom. Those are very important bonds that really stabilize the three-dimensional structure of proteins. You can have what's called hydrophobic clustering, and you can have ionic bonds too. That's what you see over here. Now, why is this important? Because a lot of what you'll learn about in your course is about how proteins can be denatured, and proteins become denatured because these often weak bonds, so this is a weak bond, there's a weak bond, there's a weak bond, this happens to be a strong bond, can be easily broken by a lot of energy, so heat, for example, or by changing the environment of a protein. That is something you learned about when you learned about enzymes, and you can learn about how enzymes are denatured. And anyhow, the result of these tertiary interactions is a complex three-dimensional shape, like what you can see down here with myoglobin, which is a protein that is in the muscle tissue, your muscle tissue. It stores oxygen in muscle. And like, you don't need to know that, but I'm trying to get rid of this. There we are. Okay, good. And finally, quaternary structure is when you have multiple polypeptide chains interacting with one another. So like over here in hemoglobin, we have actually 4 molecules, each one of which is a lot like myoglobin that I just showed you, and they interact and they form bonds with one another. And those aren't like any weaker than the bonds that are within the tertiary level. We've all just survived the pandemic. The spike protein that was on the surface of the SARS-CoV-2 virus that evolved so that it can infect us. That is another example of a quaternary protein. It's got 3 polypeptide chains that interact and form that shape that was able to bind with our cells and cause so much trouble. Protein shape, super important. Like, proteins do what they do because of their shape. Sickle cell disease, finally. In sickle cell anemia, basically what happened is it's an inherited disease. It affects about 100,000 US citizens, mostly African Americans. It affects about 8 million people worldwide, and it causes widespread complications, mostly related to improper blood clotting and inflammation. When I started teaching a long time ago, because that was like in the 1980s, sickle cell anemia was a childhood disease that killed you. And now people live into their 50s and 60s all the time because there's better medical treatment. And that, of course, happens in places where we have advanced healthcare systems, like the United States. But like in Nigeria, which is sort of the home country of sickle cell anemia, for reasons that I'll explain in a minute, the childhood mortality rate is much higher. So here's a little bit of review. The inheritance of sickle cell is autosomal and it's recessive. That means it's not on the X chromosome. It's on, I don't know what chromosome it's on, but it's on chromosome 1 through 22. And basically, you have to inherit the gene from both your parents. So here are two parents that are both heterozygotes. They have a 1 in 4 chance with every zygote that they create of passing on the disease, because each of them has a 1 in 2 chance, which means 1 in 2 times 1 in 2 is a 1 in 4 chance over here. You can see it in the Punnett square over here. They have a 2 in 4 chance, a 1 in 2 chance of having a kid who's a heterozygote, and they have a 1 in 4 chance of having a kid who's homozygous normal. The whole thing is caused by a point mutation in the gene for hemoglobin. And this is kind of astounding. Just one change, changing T to A in one spot, causes this disease, because that means that instead of coding for glutamic acid, the protein has valine in that one spot. So we're just changing one amino acid. And that is basically what I just said. The important thing is that valine is non-polar, glutamic acid is polar. So we've made a big change in the protein. The result of this is that these valines face to the outside of the molecule, and all of those anyone with sickle cell anemia will have their hemoglobin molecules interact with one another through a weak kind of bond that's called a hydrophobic interaction. Normally, hemoglobin molecules are quite shy. They're solitary. They don't like to interact. But with this mutation, the hemoglobin molecules do interact. They form these long chains or spikes, which are visible in a microscope. In other words, the cells look sickled, hence the name sickle cell anemia. And the sickling causes the cells to get caught in capillaries, which are the smallest blood vessels. And it's kind of like a fifth level structure. It's like a polymer of polymers, but the effects are disastrous, and it leads to improper clotting and inflammation. And that is really the basis of sickle cell disease. So if you know that, you're kind of set. Now, sickle cell disease is so bad. Why is it so common? And it's because the allele gives you resistance to the disease called malaria. Nigeria, it's the country with the highest level of sickle cell disease. 2% of babies are born with the disease, and between 20 and 30% of the population are heterozygotes. The frequency in the gene pool is between 10 and 15%. And why is that? Because if you're a heterozygote, you can survive the infectious disease that's called malaria. It's a protist parasite. It's injected by mosquitoes, and it infects red blood cells. But if you're a heterozygote, the parasite that gets injected doesn't do well, and you can effectively have resistance to malaria. Because of that, there's a correlation between high levels of malaria and high levels of sickle cell. And that's why the allele evolved. This is called heterozygote advantage. And there's been strong selection in the gene pool for sickle cell anemia. Folks, this has gone on for a little bit longer than I thought it was going to go. I am just going to make a commitment right now, and I'm going to say 6 p.m. 6 p.m. tomorrow night, I'm going to do another live like this. I know that gets a little bit late for folks who are on the East Coast, and I know that folks who must be in like places in Asia, because Muhammad told me that it was really, really late where he is, and I know that's even later. But otherwise, I think people on the West Coast, where I am, won't be able to participate. So we're going to do 6 p.m. tomorrow, right here. I'll do Unit 3, and what I'll do is I'll promise to throw in some FRQs as well. I just want to end by saying a couple of things, and the main thing I want to say is this. If you want to be able to effectively study, efficiently study for the next 2 weeks, please sign up for learn-biology.com. We've set up everything for you so that you can do multiple choice questions. You can do enhanced free response questions with feedback. You will learn so much. You spend 2 hours a day on learn-biology.com. You won't believe how much you'll learn. I want you to know that in addition to these lives, I'm also doing Zoom reviews that you can access by going to apbiosuccess.com/exam review. Those are not free, but I hope you have the means to sign up because they're great experiences. I did units 1 through 3 on Saturday, and I'm going to do units 4 and 5 Tuesday night. Okay, so I hope I see you there. Folks, it's been a pleasure and an honor to have you here with me. And I hope I see you again tomorrow night and Tuesday night. Everybody be well. Good luck. Study hard. You can do it. I totally believe that you can do it. You can have a great experience studying and you can crush it on the AP Bio exam. Everybody, good night. Be well. I'll see you at the next session. Bye.